Okay, 8.4, we are talking about transcription. So that is how DNA goes to RNA. So the other type of nucleic acid is RNA. RNA carries out the instructions that are in DNA. So this sounds weird. The central dogma, so another way of saying the main idea of how genetics works is that information flows one direction and it goes from DNA to RNA to proteins. That's what you see here in this picture. So DNA, RNA, then proteins. That central dogma or the main idea of genetics has three processes involved in it. Replication, which is DNA making copies of itself. Transcription, which is DNA turning into RNA, and then translation, which is RNA turning into a protein. So RNA is the link between DNA and proteins. If you didn't have the RNA, there would be no proteins being created. RNA differs in DNA in three major ways. The first way is RNA sugar is different. Its sugar is ribose, DNA was deoxyribose. The second difference is RNA has uracil instead of thymine. So it's a different nucleotide in it. So uracil will bind with adenine, just like thymine had bound with adenine. And RNA is single-stranded, not double-stranded like DNA was. When transcription happens, it's going to make three types of RNA. But transcription, once again, is the copying of DNA to make a strand of RNA. So all the three different types of RNA, which are messenger, transfer, and ribosomal RNA, are all made in this process. Transcription is catalyzed by RNA polymerase. What that means is RNA polymerase is what starts transcription. RNA polymerase and other proteins form a transcription complex. This transcription complex recognizes the start of a gene and unwinds a segment of the DNA. So that's what you see in the picture down below, is that RNA polymerase will have found the gene, unzip it, and will start adding nucleotides to the strand of DNA that it needs to copy. Nucleotides pair with one strand of DNA, so it's only going to pair with one side when it unzips it. RNA polymerase bonds the nucleotides together, and the DNA helix is going to wind again as the gene is transcribed. So as the RNA polymerase travels down it, it will be unzipping it, and then as it gets past, the DNA is going to zip back together. The RNA strand detaches from the DNA once the gene has been transcribed, and that's what you see in the picture there. So it's done, and that DNA will zip back together. Again, transcription makes three types of RNA. You have messenger RNA, which is shortened to mRNA. It's going to carry the message that's going to be from the DNA, and it carries the information on how to make the protein. Ribosomal RNA is another one, which is shortened to rRNA, and it's going to form part of the ribosomes where the proteins are going to be made. So it's going to help sort of read that messenger RNA. And then transfer RNA, which is tRNA, is going to bring amino acids in from the cytoplasm to the ribosome. So the amino acids are what make up a protein. So the transcription process is similar to replication. Transcription and replication both involve complex of enzymes and complementary base pairing. Processes have different end results. Replication copies all of the DNA. Transcription just copies a gene. So when you do it with the DNA, it's going to be 2 strands that get completely copied. In transcription, it's going to be 1 strand that gets copied. Replication, again, is going to make 1 copy. Transcription may make many copies of the thing. So that is some of the differences between them. So that is the end of the notes for section 4.